I think
All
we're good to
right.
go.
Well, okay, great. Thank you, Renee. Thanks for all you do behind the scenes as well. Welcome, everybody, to our second coregonine webinar of the month. We are still looking for a speaker in February. So I'm going to make that an effort to make that happen. But if anybody wants to make my effort easier, please reach out to Renee or me if you are interested in giving a webinar during the first week of February. Today, I'm really, really pleased to invite back again Randy Brown to give a webinar today to broaden our scope as we do occasionally beyond our Great Lakes coregonines. Randy spoke before on Bering Sea Cisco, and I'll drop the link to the YouTube recording in the chat there if that is of interest for you to go back and watch. Um, but a little bit of background about Randy. He grew up in New Mexico, actually. Moved to Alaska when he was 17 for college. That was in 1975. But after a semester, he realized, and these are his words, the woods were calling. And he moved there when he was 18 to the woods, where he built cabins, had a dog team, fished for Chinook and chum salmon in the summer and fall, hunted moose and caribou. And let's see, about 16 years later, he and his wife and their two boys moved back into the city, into Fairbanks, when he was 33 years old. And that's when he started his college career at the University of University of Alaska, Fairbanks. He then got a master's degree in 1997. There, he worked at a place called Rapids that I think he'll reference today, about 1,200 kilometers upstream from the sea. Um, and and that was also coinciding with the beginning of his career as a fish biologist with the U.S. Fish and Wildlife Service, also in Fairbanks, where he worked on migrations of salmonid fishes, looking at their critical habitats, trying to better understand their life history, using odorless microchemistry, radiotelemetry. And what he's going to share with us today is, I think, just sort of a retrospective on all that he's learned about Inconnu, their biology, their distribution, and their migration. And Randy actually retired from the U.S. Fish and Wildlife Service, I think in November of last year. We were supposed to have this webinar back in October, but of course we chose to delay that owing to the government shutdown. So we're really grateful that Randy has agreed to come now, volunteer his time, and to share this story with us, open all of our eyes and minds to this really amazing and unique coregonine. So with that, Randy, please stick around for questions and conversation later. It's 8:36 in the morning for Randy. So um, we're really excited to hear what you have to say. Thanks again.
Randy, before you get started, if you're able to move your video panel and control panel, I just noticed it's blocking some of your slides that we're seeing here.
Um, can you repeat that?
If you can, there should be a control panel probably at the top of your slides that you're seeing that shows like where you're sharing and everything else.
Yeah.
If you can just move that off of the screen, or there's probably a little caret down on the far right side that will tell you to hide controls.
Okay. Let me see if I can find that. There.
Yeah, I just noticed that when Bo was talking, I was covering some
Yeah.
of your slide.
Yeah. Well, hang on a minute. I can't see that very well. Um, I don't know how to manage that one.
If not, we can make do.
Yeah. I don't know how to get rid of that. There we go. That do it? I don't think it's going to bother the other slides. This one it took off from the title, but
Okay,
yeah,
thank you.
you bet. Hey, thanks for the introduction and the opportunity to speak today. We were going to, as Bo said, back in October and then December, and then finally we get around to it now. But thanks for showing up. This is a great image of Inconnu by Paul Vesey. I had approached him at one point because we were putting together a more or less a monograph of a lot of the different programs that gave us our understanding of Inconnu in the Yukon River, and I wanted to put an image of it, and so he said he had just done this one and made it available to me there. But I love it, and hope you do too. So when we this talk is focused on the Alaska side of the Yukon River drainage. There are Inconnu in the Canadian side in the Porcupine, as well as way up the main stem. But the research that I was involved with and others that I know was in the Alaska side. So that's gives our geographic focus of this research. I do want to acknowledge that there were a lot of people that contributed to information or publications, sampling that we use in this. Ken Alt was an inspiration, started back in the 60s, shortly after statehood, and he would go around all over the state, Yukon drainage, Kuskokwim, Northwest, sampling fish, because his job was to outline what kind of fish are where. And so he did a lot of distribution data, and that was the background to a lot of our work. In any case, wasn't just me, a lot of other people. And so some of the major topics we'll touch on today are identification of the spawning areas, migration timing, energetics and spawning frequency, and then basic life history. And spawning area, the Alatna, and there were two communities down at the mouth of the Alatna. They were actually in the Kayakuk, but right at the mouth. And they would go up and harvest fish with beach seines and gill nets and hook and line in that upper Alatna. So those were the three. There were a few others that were thought to be spawning areas, but there was no certainty to it. And so anyway, we started working at Rapids. Next slide. On the telemetry program, and with the understanding the kind of the visual landscape that these spawning areas, the known ones were. This was the Chattanooga River spawning area. It's got the Elliott Highway crossed right where the spawning area was. And in all these spawning areas, there's multiple coregonine species. In this one, there were round whitefish, Lee ciscos, humpback whitefish, along with the Inconnu. And it's this meandering river through a boreal forest landscape. Next slide. And the Selukna River was similar. It's a bigger river, but meandering river and a boreal forest landscape. And then the Alatna River, also a big meandering river, but much larger than the others. So this was our site picture. This was what we thought Inconnu spawning areas looked like. Next slide. So when we started doing the telemetry work at Rapids, we knew they were preparing to spawn and that they would be going to one or more spawning areas somewhere upstream. We used the chum salmon radio tags, and the chum salmon were, they fit into the esophageal, through the esophagus and into the stomach. And it didn't seem to bother these fish, neither the chums or the Inconnu, with the antenna sticking out. But with the tower network in place, this was a reasonable first approach of this. We were a little bothered by it because we didn't know if the Inconnu would regurgitate them and spit them out at some point so they could feed normally. We didn't think they could pass it through the stomach and the intestine, but we didn't really know. But it was where we were at that point in time. Next slide. And this is the nature of that upper part of the Yukon when we were doing this work. Rapids, you can see it's a little upstream of the mouth of the Tanana on the center left part of this map there. And there's these little towers icons there, and those were radio receiving stations that were in place at the time. And it's about 500 kilometers from the tower at Rapids to the tower up near Circle. Next slide. So we thought we could narrow it down to a location, and we actually flew all these different Clearwater rivers that flow into the Yukon during that in that range, and we thought that it was going to be something like the Selukna or the Alatna, but not to be. They went and spawned in the upper part of the Yukon Flats. There's about 120-kilometer stretch of river with main bank to main bank being 3 to 5 kilometers wide, with all these different channels and islands scattered all throughout. All of the radio tag fish that we put in the river there came up into this spot. And we ended up doing beach seining and electrofishing eventually. And there's broad whitefish, there's humpback whitefish. Bering cisco is probably the most numerous of the coregonids in this area. And round whitefish, least cisco. And then all the egg eaters, and then the predators that eat these smaller fish. It's just a massive fishery area that we had no clue about before that. Next slide. We did eventually graduate to surgical implants using clove oil for a while, and then aqueous as the anesthetic. And we were able to put these transmitters in. They were about the size of your pointer finger, and they would last for about 4 or 5 years. And you could program them for on and off periods as well, if you wanted to. And so these ones allowed us to see multiple years of movement of these Inconnu. And it really opened the landscape for us in that regard. Next slide. Similar to the Yukon, where we had rapids catching these fish preparing to spawn in the lower Tanana, they were also catching coregonids and that were preparing to spawn. And so I worked with some of my colleagues at Fish and Game and Fish and Wildlife Services. We put radios in Inconnu and broad whitefish and humpback whitefish down in the lower Tanana, and they came up to this braided region of the Tanana that looks quite similar to that one in the Yukon Flats, except it's smaller. It doesn't have quite the breadth or length than the Yukon Flats one, but it similarly is full of fish in the fall. Next slide. And then we found one other spawning area, which was in the upper Innoko. We had a big tagging program in the Innoko for a few years, and we were of the impression that they did not spawn in the upper Innoko, but we did find it in this one place. And there's this long pool against this one bank there that it looked like all of our radios were in there. We did make a trip up there to verify. It's a 1,000-mile boat trip from the Hall Road Bridge down the Yukon and up the Innoko to this site. But it gave us the ability to inspect it and fish. We got genetic samples from 100 fish right off the bat, and we're able to verify that that these were in fact preparing to spawn. Next slide. So at the end, we had 6 definitive spawning areas: the Yukon Flats, the Tanana Flats, the Chattanooga, the Selukna, the Innoko, and the Alatna River. And those, none of our radios from any of the radio tagging project went anywhere else. If they went somewhere to spawn, it was one of those places. We had towers out along the river, so we were able to monitor them moving upstream and downstream in various places. But this is what it looks like. It's possible that there might be a really small population somewhere that we're unaware of, but we don't think that's really the case. Next slide, please. We then went into the otolith chemistry, and they're not all exactly in the right timing, but we did a lot of otolith chemistry because the 2 parts of having these Inconnu at rapids was where do they go to spawn, and then where do they go when they're not engaged in that part of the river. And so we did some otolith chemistry to see whether they had been to the sea, and we used a transverse section. Next slide. Which we bound to a slide, and then examined with a compound microscope and transmitted light. And there were also these polarizing filters on this microscope so that you had a black background. And you can see the arcing lines on that right side of the sulcus with the white spots on them. Those were the points in which we judged that to be an annuli. And this is a 29-year-old fish. That's what we judged it to be. It's a really nice, clean one. Next slide. The oldest Inconnu that we've ever seen was one that my colleague Bill Carter found up in the Selawik River. And and he prepared this slide, but it is, we judge it to be 41 years old. It's an amazing fish. We haven't found any that were that age in the Yukon at this point. Next slide. Once we did have ages, we had good population level ages from the Alatna River, the Yukon Flats, and the Selawik River, and we put them all together in this growth profile here, the von Bertalanffy. And one thing that's really clear is you have pretty much rapid growth to spawning maturity or a little farther, and then they just lay down, and not very much length comes with increased age there. And you can see the icons at the far end of the far right side are up there around the 25 to 28 years old, and mostly females than the really old ones. Females definitely grow bigger than males. Next slide. So part of the otolith chemistry work was to look at these Inconnu and identify whether they had been to sea or not. We did a paper on that back in 2007, but we sampled fish from all different parts of the middle and upper drainage in the Yukon. And all of the ones that were up in the upper Porcupine or the upper main stem near the border were like this top profile, with the strontium calcium ratios being quite low, and definitely not going to sea. None of those fish were going to sea. The Yukon Flats population, though, all of the ones that we tested were had been to sea, at least some. That's the center profile there, with at least 3 different trips to sea as a younger fish, and then it looks like that fish came into freshwater and didn't return to salt. And then in the bottom profile, it looks like that fish has been to sea every year through its life and back into freshwater, which is consistent with some of the telemetry data that we have. And I'll share that with you a little bit later. Next slide, please. So that lower profile where that fish is going to sea every year, we create these strontium density maps with the electron microprobe. It's got a 10 micron spot size for these, and it creates a grid pattern across it. Doesn't have to shoot the beam at the thing for very long, and it gets the gist of the strontium density across the otolith there. But you can see in this one, the yellow is the high strontium, so they've been into sea during those periods of time. And then the orange is freshwater. The core over on the lower left is where they're spawned and spend a few months before they actually get to sea. And then from that point on, it looks like they go to sea and come back into fresh every year. Next slide. So one of the great things about rapids is as that mark recapture program kept going, we realized that having those fish in the live boxes for lengths of time was not a good thing for the fish, and wanted to be able to just let them go through the wheel and leave the door open on the back of the live box so they could swim out. And so they developed this camera system. It was a motion activated camera. You can see the white encasing of it. And we had a computer out there and a battery that was being charged by a water turbine under the fish wheel raft. It was a great system. And in addition to being able to identify the chum salmon, next slide, which would go through the camera system, you can see the the spaghetti tag in it. What we were doing was week-long population estimates based on the weekly output of tagged fish. And so we would have that spaghetti tag was a different color each week. And this thing was developed by a guy, Dave Dahm, one of my colleagues there, who was colorblind, completely colorblind, but he he was still able to tell these tags from one week to the next. And if he could do it, then anybody could do it. Next slide. But in addition to the chum salmon and the tagged versus not tagged fish going through the recapture wheel, as well as fish that were recaptured in the original tagging fish wheel, we would catch images of all the other fish as well, like the burbot. Next slide. And the Inconnu. This Inconnu looks like it's screaming, but of course we didn't hear anything. But it gave us the opportunity to actually do run timing data there. We had 15 years of data. Next slide. That went along with these run timing profiles. So the rapids there is it's actually a percent of the total number of Inconnu going through in the 15 years, day by day. That's how that run profile comes to be. And you can see that those, because we would run that fish wheel from early summer all the way through into October, when it would start to get iced up. And so Stan Zaray he would collect the data for every day and count out all the different species. And so in this case, it's the run timing of the Inconnu. Starting in August, they start really ramping up, and a lot of them are moving through that fish wheel on their way up to the Yukon Flats. The blue profile there is the run timing of Inconnu that spent the winter down in the Delta and the estuary that are coming back into freshwater. Those were collected. That was a 29-year data set that the Alaska Department of Fish and Game collected from their pilot station sonar operation, 195 kilometers upstream from the sea. And so what they do is they had sonars on both sides of the river, and they had all these different species of fish going through. So they counted just fish on going upstream, and then they had a netting program that they could use to proportionalize the total number of fish going upstream into the different species. And these were the Inconnu. And so you can see that they start coming in at the late May and run really strong through June, and then taper off. It's not that they're not there. They do keep going through the summer, but at a much lower rate. And so these gave us some real insight into, the timing mechanisms and what these fish were doing along the river. Next slide. And in addition, we started getting, with the telemetry program, we started getting run speed, swimming speed upstream. We had 52 fish that had gone by 2 or more of those remote stations that were several hundred kilometers apart for these data. And so you get a mean upstream migration rate of 22.5 kilometers per day. Downstream was considerably faster, as you might expect. And we had 58 samples that were going downstream after spawning, and with the fastest one being almost 200 kilometers in a day, which isn't really a surprise for going down. But they turn around after they spawn and they head downriver to the Delta, mostly. Next slide. Those ones that come in through Pilot Station in late May and early June, most of those fish are coming into the Innoko River to feed. We had a bunch of radio tags in the Innoko for a number of years, and they would come in shortly after that big pulse coming through at Pilot Station, shortly after it started. So they would start coming in late May and through June into the Innoko. And then those that would spawn would come out. We had a tower in the lower Innoko. So when they would leave, we would know it. And it's this giant flat water system. And right here, the main Innoko is on the left side, flowing towards us, and the Iditarod River is on the right side, flowing towards the Innoko. The Innoko there is about 300 meters wide and about 15 meters deep. And it's just a stunning piece of waterway. And these Inconnu and other fish come in there in huge numbers. There's ospreys all over. And so they come in there to feed in the summer, and then they head back out onto the river and migrate up the Kayakuk to the Alatna and the main stem all the way into the Yukon Flats. And those are the majority of fish, of our radio tagged fish from the Innoko, went to those two places. Next slide. Another place that they go to when they come into the Yukon and start migrating upstream are the mouths and lower reaches of many of these clear water streams, and they can be caught in that mixing zone. This is the Zitsiana River, which comes from the south and flows into the Tanana. This is a back channel of the Tanana River there. But in almost all of these situations, you can go in the summer and there'll be a few Inconnu there. Not huge numbers, but they'll be there. And one of the interesting things with the radio telemetry was when we would catch fish in these places and tag them with these long-term tags, they would be right back in the exact same place the next year. They remember everywhere they've been, I think. And I don't know how they find these places to start with, whether it's kind of a random selection, but they go right back to the same places. Next slide. So Lambert and Dodson, what a paper this was. It had a lot of influence and still does for people. They were looking at lake whitefish and lake ciscos, anadromous forms, and they did the energetics for them and decided that there was really no possibility that they could spawn 2 years running. And it kind of permeated the thought with the northern coregonids, this idea that they could actually get enough nutrient and energy over the course of a winter and early summer to make another go of it the next year. Next slide. But and there is no question that there's a big energy decline when they do go to spawn. These are data that John Gherkin and got on the Selukna River, and Dave Essie got also on the Selukna, but downstream from the spawning migration. Dave Essie did a sonar account of the outmigration 2 years running, and he collected a number of post-spawning Inconnu, partly to verify that it was Inconnu he was counting, and partly to verify that they had spawned. So the pre-spawning fish are in that upper curved regression. The white icons are females, and the yellow icons are males. Males tend to be smaller. And then the blue line is from Dave Essie's work downstream, post-spawning. And so you can see that, say, if you were to look at a 90 centimeter long female, you'd see that there's a loss of about 3 kilograms between the two, the pre-spawning and post-spawning aspects. And similarly, if you go up to 700 millimeter long male, the difference between those two regressions is about 1 kilogram, and probably mostly gonad. But if these fish were going to spawn a second year, they'd have to replace that energy in order to be able to do it again. And we still don't know how much energy is drawn from them for the migration. If they're down in the lower river and they migrate, 1,000, 1,600 kilometers up the river to get to the Yukon Flats or the Alatna River from feeding areas. We just don't have those data yet. But unequivocally, it's going to take an important feeding expedition in order to be able to spawn on consecutive years. But I think that I think they can do it. Next slide. So one of the interesting things was that in these three big radio tagging programs, one down in the Innoko, 115 fish, one in the Newitna, so that's 60 fish, and one in the Tanana with 54 fish radio tagged. So these are long-term radio tags, so we have the ability to see how frequently they spawn. And you notice about a third of the fish from each of these didn't spawn at all in 4 years. And of those that did spawn, most of them spawned once. And there's a few fish that spawned 2 times, 3 times, and 4 times, more from the Innoko than the other 2 spawning populations. But that is still, and why they don't spawn in 4 years, I don't know. But similar, you remember that Zitsiana coming down, and I'm wondering, wonder how they find these places. They may find places that they go back to that are not really very productive, but they may just not go somewhere else. They may not be, prospecting for better places. I don't really know, but I think it's really unusual that they're going to be spawning 0 times in 4 years. Next slide. We did get a number of fish that spawned on consecutive years from all of these populations. 2 times was recorded by 15 fish from the Innoko, and 5 from the Newitna, and 3 from the Tanana. Now the Innoko fish are primarily anadromous. They go down to sea. We've done testing in the Newitna, and very few go down to the sea, and the same in the Tanana. There are some, but not very many that go to sea, go out into the estuary. We did have 7 fish from the Innoko spawn 3 times consecutively, and we had 5 fish from the Innoko tagging group that spawned 4 times consecutively. It really is stunning that they can do that. But here we'll get into the reason that I think they do. And in truth, what we did was find them on the spawning grounds and know that they left and went downstream afterwards 5 different times or 4 different times for that Innoko River group. And so, but we have sampled lots and lots of fish on spawning areas in several different places, and we just never find non-spawners. And so it's a judgment that if they are present on the spawning grounds, our sampling work suggests that they are in fact spawning. So that's the nature of this particular table. Next slide. So a few years ago, I had been doing work up in the Selawik National Wildlife Refuge. So that's Northwest Alaska. And a couple of enterprising high school students, Grant Magdans and Taylor Everett, they contacted me, and they wanted to have an idea for a fisheries project for a science fair exhibit for them. And I think that fish investigations are really kind of challenging, and I didn't have much hope for it to start with, but I said, well, you know what? You have access to Inconnu up there, and there's 2 big populations of Inconnu up in that area. And I said, we don't know whether they're feeding in the winter, and it would be great to have a study on whether these fish caught in the December and January could be feeding. And they took it on, and I told them, you can't get fish and just take the big ones that have giant bellies. You got to have some unbiased way of selecting them if you have more than what you're going to sample. And so they had a friend who had a dog team and hung a net under the ice in the winter, and they would sometimes get 40 or 60 Inconnu overnight. And so they Grant and Taylor came up with this idea. They're going to line them out from smallest to biggest. And if they can deal with a third of the catch, they would take one every third fish. And I think they really nailed it there as a good way to go about being unbiased. Next slide. So this is that region there. It's a really unique place. There's a big spawning populations of Inconnu in the Kobuk River, which is the upper river flowing from the right side over to the left side, and the Selawik River, which is farther south, and then it runs from the left side of the image over to the right side of the image over to the left side in Selawik Lake. Too. Selawik Lake is clear. I mean, it's a freshwater, and Hotham Inlet, that much bigger water body, is brackish water, and it's saltwater out in Kotzebue Sound. So the Inconnu can't really go out there during the winter because the water is too cold, and they don't have antifreeze capabilities. But they do go to the northern side of that Hotham Inlet, and that's where these fish were being captured. Next slide. And this was one of the big Inconnu they caught that had a bunch of Pacific herring in its stomach. And while not all fish that they caught had food in their stomachs, a considerable number did. And they would also get saffron cod and rainbow smelt up there. So there was that mixing zone where the marine fish that did have the antifreeze capability would meet the freshwater fish that didn't. And these Inconnu were definitely gorging on these small but very nutritious marine fish. Next slide. Now this is an image of the Yukon Delta, and it's about 50 kilometers apart from the south side of it to the north side of it. And there's several different distributaries in it and several communities. There's 3 on the south channel, which is the biggest of the channels, and 1 on the north side. And they fish for Inconnu and other whitefishes in the winter under the ice, and they catch them. So we know they go down there, and the rainbow smelt and saffron cod and Pacific herring are also present down there. So we think that there are fish that spawn on consecutive years, but they probably have to go down into that part of the river in order to be able to feed. One of the things that we did with the Selawik River population was we put radio tags on them, but they didn't go down. We didn't have them go down into the Delta. They were much more wintering in the lower Newitna and then in the Yukon, just randomly behind an island or out in the middle of the river. And they would be there in October and in January and in April. And I had this vision that they've got their pelvic and pectoral fins in the gravel and their mouth open facing upstream. And I think that's how they spend the winter without eating. And those ones would be much less likely to have the spawning on multiple years in a row. Next slide. So these, there were 3 populations for which we had age data sufficient to estimate survival rates. The Alatna River and the Yukon Flats have a large fraction of their population that go down into the estuary during the winter. And the Selawik, as I mentioned, really doesn't. There's not very many of those Selawik fish that go down into the Delta based on otolith chemistry and the telemetry work that was done. There's a really stark difference in survival with the Alatna River having 42% survival from one year to the next, and the Yukon River population seems to have a survival of about 63%, and the Selawik, though, it's up towards 84% of their the adults survive from 1 year to the next, and it kind of gives you a different perspective of life history attributes. You know, the selection pressures on the Alatna and the Yukon are such that they go down there, they're subject to a fishery there. When they come back into fresh water and they start migrating upstream, they're subject to salmon nets and fish wheels. So they're being harvested. There's a harvest issue when they go down that far and then make it back up to their spawning grounds. Whereas the Selekna fish, they really don't have that the fishing related mortality because they're not active in the Yukon during the summer, at least not most of them. And in the lower Newitna and up in the Selekna River, there isn't a fishery going on them other than the casual boater that might throw a crocodile or another spoon out and catch one here and there. And so if you look at lifetime production, the Selekna fish are probably not going to spawn on consecutive years, except very rarely, whereas the Yukon Flats and the Alatna will be spawning on consecutive years, at least some of them. And so if you look at lifetime production, I mean, the Selekna might actually spawn more times just because they grow a lot older. Anyway, I think it's an interesting phenomena in this mix. Next slide. That's all I got for you today. Thank you very much. It's unfortunate it stalled us a little bit, but I hope you enjoyed this.
Thanks very much, Randy. That was great. Fantastic. Trying to think about what question I might ask you because you covered so many different things about their ecology. Maybe just one quick question would be, compared to your knowledge when you started studying this fish that we didn't know a lot about to today, like what was the most surprising or what finding are you maybe most proud to have sort of uncovered with your colleagues over the past 30 years?
Mm-hmm. Yeah, well, thank you for the question. And I think that understanding the sequential year spawning, the consecutive year spawning, with some of them doing it several times in a row. And that's really reasonably stunning. And I think that we've had this perception that doesn't happen. And before these long-term radio tags, we really didn't have a way to look at that. Mm-hmm. And so I think that was probably one of the more unique aspects of the program.
Right.
And the aspect that Dave Essie brought to the mix with the post spawners and the potential for understanding how much nutrient and energy they expend during a migration and spawning event. Yeah. We haven't really looked at that before either. So anyway.
Yeah, the variation in the skipping and then some able to do it consecutively. Sure. Very, very cool. Also, those GSI levels are just kind of visual image there of how many eggs, nearly 500,000 eggs and 30% of the weight of the female. But yet females still seem to have survived. I mean, just looking at like your von Bertalanffy curves to still grow larger sizes and to live longer ages.
Yeah.
Despite that huge investment is another really interesting thing, I think, too, compared to the males. What were the male GSIs? Do you remember like what sort of scale they would be? More like the 5 to 10%, or?
The Freshwater Institute, Kim and Melanie Van Gerwen-Toyn, they've toyed with that, and then also from the data of Lambert and Dotson, the male GSIs are about 1 order of magnitude less. And the reason I don't do them is because it would require a different kind of a weight measurement tool in order to
be sensitive enough. Yeah.
To be sensitive enough to make it work. But they are about, 10% of what the females produce. So it'd be challenging to get it, but they did it, and it is, but it's not a very steep curve.
Yeah. So I don't know if you can see messages in the chat, Randy. People are saying it was worth the wait. Excellent presentation. Great story. Thanks for sharing. Um, and certainly people were giving you lots of claps through sort of the cartoon there.
I saw that.
Let's
Yeah, I
open it
don't
up.
see the chat, but yeah.
Okay. Well, let's open it up to questions. People can either turn on their cameras, ask it, in person, so to speak. Scott Hansen in the chat asks what sort of inferences you might have on how they're making their spawning habitat selections. I mean, you talked a little bit about sort of those winding rivers, but then you also sort of showed the flats, but maybe more about maybe some of the specifics with respect to substrate, perhaps, or flow, or I don't know.
Well, I had left out a few things in the presentation just because it was getting a little too long, but I do a lot of thought on that. And I think that John Gherkin did some real study on that in the Selukna River. He spent, I think, 3 years doing work there. And one of the things he noticed, he did sampling work to understand not just the upstream and downstream bounds of that spawning area, but where the fish sit when they're in it. And as they hold, they're in the pools, and then when they go to spawn, they go up into the runs where you have gravel and cobble, and it's cleaned off to some extent, and that's where they spawn. So that these eggs are going to drift to the bottom and get stuck in crevices and various other things, so that the egg eaters don't get them. And also one of the features that he was able to discuss was the conductivity. He thought that the conductivity in the Selukna was greater than many of the other streams in the area, and that might have something to do with it, given the relationship between conductivity and groundwater coming into the system. And Lisa Stubbe, who did work on the Kuskokwim River Inconnu populations, indicated that the spawning population she was able to find there also had relatively high conductivity. And I think there was a gal, Lisa Worth, did work on the Tanana River flats there, because there's chum salmon that spawn in the same places that the Inconnu are present, Inconnu and other coregonids. And she was able to document that there is that groundwater flow that comes into that system and makes it so there's some sort of warm water there. And I know Kim Howland and her colleagues did work on temperature days that required for hatching and then for growth. And so that was a really interesting piece of the equation as well, because they aren't going to hatch if they're down at zero all the time, which the flow of the river between the gravel and the ice can be pretty darn close to there during the winter. So
that is really
the
critical.
upwelling might be a big important issue.
So kind of related to that, I mean, I thought that another interesting part of your talk was all the coregonine species that use these habitats and actually were found spawning in the same general area.
Same place.
It sounds, And so how are they like maintaining different, like the reproductive isolation, especially if all of them see this advantage of these groundwater sort of warm refuges where their eggs or embryos could incubate faster? Like, do you all have a sense of maybe is there timing where the different 5 or 6 species spawn at different times, or is that not really known yet?
I think I don't know exactly, but I don't think they avoid each other. I think they just I mean, there may be subtle differences in habitat that they pay attention to, so that there's one species over there and one species over here, depth, or I don't think they're picking something because of substrate necessarily. It's more the run of the river or something. The Yukon Flats is a wild place because you've got it's really porous gravels through that region, and so I tend to think that that these big islands have water being subducting under the island during the summer, and it slowly moves through and comes out the bottom side. So it's a different temperature than the water in the flow of the river. And you have these open leads that are along a lot of the different islands. And so I think that some of those eggs would go down and be entrained in that area
Wow.
where it might be a little warmer. And so they're,
Yeah.
you know, even if it's just a degree or two warmer, it's going to make a difference in when they hatch and when they go. So
Very cool. There's another question in the chat from Dylan Undland, who asks if you've observed straying of individuals between spawning sites or tributaries within the Yukon drainage.
we had, that's a good question. We had one fish that spawned in the Yukon Flats one year, and the next year spawned in the Alatna River.
Oh.
And another one that had spawned in the Alatna River one year and then went and spawned in the Yukon Flats. So those two are they're both about 1,600 kilometers from the sea, and yet there's that one pivot point right at the mouth of the Kayakuk where you make one decision or another. So I tend to think that they probably got along with a group of fish heading upstream, and they could smell it. They aren't seeing anything in the Yukon, and got sidetracked, but went anyway. So there's a small amount of straying anyway in that.
Oh, right on cue. We have a geneticist question.
Uh-oh.
Wendy, and you hosted Randy before.
Yeah. We've met before. Actually, it's not a genetics question,
Oh,
amazingly.
well, I had
Um,
one. I thought there would be. So yeah, go ahead.
Well, we are starting some genetics on them, so
Mm.
might have questions later. I guess just because I am starting to work on the system, one thing that is sort of being presented as a concern by some of the communities that utilize the Inconnu on Great Slave Lake, which is part of in the range, but in Canada, is this long migration they do along the Mackenzie River is becoming a little bit more treacherous due to you know, it's getting warmer faster in the spring and also getting shallower faster in the spring. And I was just kind of wondering, in any of the big river systems you're working on, is this something that you're starting to see as an issue?
Mm. Well, thank you for the question. We do consider the temperature, and Vanessa von Bila has been focused on that with more with the salmon, and did a paper I think in 2020 on all the different heat-related die-offs of salmon. I don't think we've seen that with the coregonids yet. I mean, there are times, you know, in that Innoko area, you could see those big giant lakes. A lot of them have these little windy streams that go out to the bigger rivers, and humpback whitefish, least cisco, and broad whitefish will make their way into those to feed, and they sometimes have die-offs in those lakes, but that's a very different situation. In the main river, I haven't really noticed it or seen literature on it with the coregonids. So I don't know. It probably, if it keeps getting warmer, it probably will be an issue, but I'm not aware of it.
Thanks, Wendy. So Randy, I'll ask a probably very naive, maybe dumb question. The Inconnu obviously are piscivorous. We do see some piscivory of coregonines in the Great Lakes, but it's relatively rare. They're more like some of the smaller coregonine species that you talked about. At least for the ciscos, you had the least bearing. I can't remember the bearing, and then the Inconnu. Of course, Inconnu have a stenotus genus. Like I'm just wondering how such a piscivorous, fast-growing coregonine potentially evolved along with all these other large salmonids in that system. Do you have any insight into sort of the uniqueness of the Inconnu in terms of its origins, or?
Mm. Well, I know it hybridizes with several species. You know, it is known to hybridize with humpback whitefish wherever they're spawning together. I don't think they pair together, but they're present spawning in the same kind of places. So Ken Alt did a paper on that long time ago, and Jim Reist and some of his colleagues did that over on the Anderson River, I believe, Lower Anderson, maybe Lower Mackenzie as well. I forget where all they were, but they were able to show that there was hybridization for several of the different paired samples there, paired species. And I think that it happens on a regular basis. I think anywhere in these spawning areas where you have both. I don't think they're all that different genetically, the Inconnu with the Lee Cisco, or the Inconnu with the humpback whitefish.
Interesting.
Broad whitefish is a little different. They spawn really late, and I think the likelihood that they would have an accidental breeding is low. Whereas the humpback whitefish, Lee Cisco, and Inconnu, they're all spawning kind of synchronously. So we do see hybrids, and we worked on a paper not too long ago with Kevin Fraley and some others that focused on hybrids and traditional knowledge of hybrids out in, mostly they were dealing with Northwest Alaska, but they're present.
Yeah, James Savary just dropped something in the chat. James, I don't know if you want to talk about like where that is geographically, or.
Yeah, I was just showing a picture of the hybrid we caught when we were doing radio telemetry work on the Kobuk years ago, and Randy is the one who did. Sorry, I was probably muffled there.
No, we heard you.
Randy is
You're
the
good.
one that did. He trained us up on how to do all the surgeries and was involved in that project as well. So when he
Nice.
mentioned it, I just thought I'd show people a picture. And Randy and I have talked about sheepfish quite a bit over the years, and on the telemetry project that we did on the Kobuk, we did see, consecutive spawning with males and with females. And we had one girl that came back 4 years in a row, and we talked about their diet being a major part of that. And I think there might be an age component too. Part of me feels like that girl might have been in her prime years of spawning, maybe more so than someone that's like 25 years old. And great talk, Randy. And just as a side note, we call Randy the Jedi Master.
Thanks, James.
Thank you so much, James. Yeah, that's great. Yeah, fascinating system. All of these coregonines, hybridization, all these unique tributary, unique places where they spawn, but obviously some straying. Sounds like Wendy and some others may be doing some genetics work on these different spawning locations if it hasn't happened yet.
Yeah, Jeff Olson with the genetics lab of U.S. Fish and Wildlife Service, he's got all the samples that we've collected. He came out on that trip out to the Anoka to sample the fish as well. But that 1,000-mile boat trip, he didn't go the whole 1,000 miles because we picked him up in Shaglik, a community in the lower Anoka. But it was what a great trip. But I know that he routinely shares genetic material with somebody doing research. So yeah.
Do you have any good grizzly bear stories to share for all of us that don't see grizzly bears very often?
Oh yeah. So I'll tell you one story, okay? We were living down in Cold Bay. So Cold Bay is way out the Alaska Peninsula on the south side. And there's no trees. and Eisenbeck Lagoon is in the north side of it, and it's a great lagoon, and there's a lot of different fish streams that go up on the south side as well as the north side of the peninsula at that area. And my wife was teaching out there, and I was working or not working different times. And this one guy, I was working with the power company down there, Gary Ferguson, and they were saying that the high school kids used to walk up on this mountain right behind the Coal Bay, the mountain that you'd go to if there was a tidal wave coming your way. And on the backside, he said there was this cave. And so I thought that the high schoolers used to go to and smoke pot. And I said, well, we had two kids. One was in junior high, one was in elementary, and then the refuge manager had a boy in the same age class. And we decided we're going to go up with our dogs. I had two trapline dogs that were going to go with me, but I wanted to go and see the cave, take them on a walking trip to see the cave. So we get up on this little bluff, and we can see the cave, and there's fresh dirt out in front of it, and there's brown bears all over the place there. And so I had the kids sit on this one cliff that he could watch down there, and I went down. There was a ledge along the base of this cliff over to the cave, and I was confident. I had a lot of confidence in my dogs. You know, they were big dogs, 120-pound huskies, and that they were going to give me an alert if there's a problem with the cave. But I thought we'll go up, but if there's a bear in it, we won't go in. You know, but the dogs should be able to tell me. So they go walking by this pile of dirt without a second look. And so I went up. I had a rifle, and I peeked around the corner, and here's this big brown bear about 20 feet back in this thing, sitting on his butt, and he stood up when I peeked around the corner. And so I turned around and started running back on this little ledge and put a round in my rifle, hauling ass. And the bear never came out. And I get up there with the kids, and my older boy, he says, "I thought you told us you don't run from a bear." I'm saying, like, well, that one was a cave. There's only one way to go. So there it is. Those bears down there were not particularly aggressive towards people. They walked through town, and they just they had all the fish they wanted. And so anyway.
Thanks for sharing that. No, that's great. Wow. Thanks for helping us see the other side of what goes into getting all these data too. I mean, the difficulties and the travels and the boat trips and the float fly-ins. I'm sure all of that has been a part of your career. All right. I'm not seeing any other questions in the chat, or last chance for anybody to flip their camera on and have a question here for Randy. All right. Well, if not, Randy, we had a tremendous turnout. Almost everybody stuck around despite the technical difficulties, for which we're really grateful. Thanks again for volunteering to wake up in the morning, do this talk early as a newly retired Fish and Wildlife employee.
There we go.
It was great for all of us getting to learn much more about Inconnu. So
thank you. Appreciate the invite.
Thank you.
Yes,
All
thanks,
right,
Randy.
we will be in touch. Yeah, we'll be in touch about a February meeting, February talk, folks.